Okay, hi everyone. Good evening. I'm so excited to be here with you all. My name is Dr. Jacinda Abdul Mutakabir. I'm currently an ambassador for Pharmacy Initiative Leaders, and we're so excited to have you all join us here for this conversation. So just a bit of background on pills. We are dedicated to empowering underrepresented individuals of minoritized groups as they matriculate through the pharmacy profession. Pills has a very distinct culture that includes leadership, authenticity, vision, innovation, as well as diversity and inclusion. So we want to thank you all today for just joining us as we propel our mission and vision forward. And we have this conversation detailing the COVID-19 vaccine and hesitancy in the Black community. So as I previously stated, my name is Dr. Jacinda Abdul Mutakabir. I am currently an assistant professor at Loma Linda University in Loma Linda, California. I trained at Howard University. I went to Howard University for undergrad as well. I graduated from the University of St. Joseph, where I obtained my doctorate degree in pharmacy. I, as I stated, completed my pharmacy residency at the Howard University Hospital. And then I went on to complete an infectious diseases research fellowship at Wayne State University, as well as a master's in public health. So as a Black woman in pharmacy, as a Black woman in infectious diseases, I've had a very unique and distinct pleasure and honor to have come into contact with just really amazing women. And we were able to forge sisterhood. So here you all see this semblance of my Avengers, my Black women pharmacists. So we have had the honor of growing together in infectious diseases, and through us growing together, we've had a unique opportunity of growing together through a pandemic. So noting that 1 in 875 Black people have died from COVID-19, and noting that 18.7% of African Americans account for the COVID-19 deaths, while we as African Americans only make up 12.5% of the entire United States population. So we are disproportionately affected by this virus. So my girls and I, we noted this and we thought to ourselves, how is it that we just increase awareness, that we show empathy, that we also explain and elaborate on institutional distrust, but we make sure that our people have the education that it is that they need to make informed decisions on whether they are to receive the vaccine. So essentially, how do we deliver information for us, by us? So with that, we were privileged to be able to use the Pills platform to go ahead and have this conversation. So I will kind of step back from voicing too much, and I will kind of route you guys in what we're going to talk about as we get more into the evening. So we will first begin with describing previous pandemics and how those previous pandemics have kind of gotten us to where we are with managing COVID-19. We will then talk about vaccine development. What does vaccine development look like? Then we'll talk about the Tuskegee syphilis study. So what exactly is it that the Tuskegee syphilis study, what was it? And what does that look like? And what are the implications of that Tuskegee study and how African Americans are approaching the COVID-19 vaccine? And then finally, we will elaborate more on just the COVID-19 vaccines themselves and how it is they are constructed, and what do they look like, and what do the studies and the clinical data that we see on CNN, what does that mean? So as I said, I'm going to take a step back and I'm going to go ahead and let my conversationalists introduce themselves. I also want to let you all know that as of right now, you all are muted. My apologies. So if you have questions, you can ask them through the chat feature. Various Pills ambassadors, as well as myself, are monitoring that chat feature so that we will make sure that we backtrack for any questions that are concerning the points that we are discussing are answered. So we want to make sure that we are able to address your concerns, but that we are also able to stay on task for the conversation at hand. At the conclusion of the conversation, you all will have that opportunity to go ahead and voice your questions verbally. So we'll start with you, Dr. Christian, and if you could just introduce yourself so we can get the conversation underway.
Awesome. Thanks, Jacinda. Thank you, Pills. Hello, everyone. So my name is Robbie Christian. I am a clinical infectious diseases pharmacy specialist at Our Lady of the Lake Regional Medical Center. It's here in Baton Rouge, Louisiana in the South. Born in Cleveland, Ohio, though. I completed my PGY1 at the Miami VA, PGY2 at the Cleveland VA, which I missed so much and got a great experience there. I also wanted to let everyone know that I have not yet received this vaccine. However, I am scheduled to receive it on January the 4th.
Hi, everyone. My name is Yawande, or Dr. Dio, and currently my role is a clinical pharmacist, is specialized in infectious diseases. I am currently in the process of transitioning to a different role or to a new role at a different hospital. In terms of my education, I went to pharmacy school at Howard University in DC, and then I went on to do my first year of residency training at the VA hospital right there in DC, and I did a second year of specialized training in infectious diseases at the VA in Houston, and I've been working since last year as an infectious disease specialist. As regards to the vaccine, I haven't gotten it yet, but I do plan on getting it when it is offered to me. Thank you.
Everyone, my name is Michelle Maxim. I am an infectious disease clinical pharmacist at Jackson Memorial Hospital in Miami, Florida. I completed my undergrad degree at the University of Georgia in Athens, Georgia. I went on to do pharmacy school at the Medical University of South Carolina. I did my PGY1 at Wake Forest Baptist Health in Winston-Salem, North Carolina, and completed my ID training at Grady Health System in Atlanta, Georgia. I did get my first dose of the 2-dose schedule, the Pfizer vaccine, on December 16th. I am still here, doing well, and I'll be getting my second dose January 6th.
Okay, and my name is Brenda Simiyu. I am an infectious diseases antimicrobial stewardship pharmacist at West Jefferson Medical Center here in New Orleans. I did my training at Wingate University. I went on to do a PGY1 at Jackson Hospital in Montgomery, Alabama, PGY2 at the Carilion Clinic in Virginia. And I also did get the vaccine on December 14th, and I can't wait for my second shot on the 7th of January.
Thank you, everyone, for introducing yourselves. And also, what I didn't state, I also received a COVID-19 vaccine. I am excited. I am still alive in the flesh. Amen. I received my vaccine on December 17th. And December 18th, and I will, or was it was the week after. So the week before Christmas. And I will be getting my second dose of the vaccine, the Pfizer vaccine, on January 8th. So it was 21 days prior to January 8th. So as Michelle said, I am still alive and well. And yeah, so we're excited to go ahead and get started with the conversation. So I think the biggest thing for me has been how kind of aligning and kind of addressing how previous pandemics have really played into how it is that we've dealt with this pandemic we're currently in, you know, with the mask wearing and everything of that sort, and socially distancing. I remember that being a big thing with the COVID-19 outbreak. So how exactly did we get there? Michelle, if you could kind of lead us into that aspect of it.
Sure. So there have been several, pandemics that we've experienced, unfortunately. And the 1918 influenza, or the Spanish flu, was really one of the first pandemics that kind of gave us a lot of information on how to effectively manage with non-pharmacological means. So essentially, it was first identified in military personnel in the US. About 500 million people actually were infected. 50 million of those patients actually died. And of those 50 million, 675,000 in the US passed away. So the mortality rate they found was that it was actually in the extremes of ages, the higher mortality. So patients that were less than 5, patients that were 65 years or older, and then oddly enough, there was higher mortality in this 15 to 40 range of what we called healthy patients. And so around that time, if you can think back, the American World War was going on. About 30% of physicians were actually deployed. And so around that time, they weren't able to test for the virus. We didn't have antibiotics. Penicillin wouldn't be discovered for, 10 years later. There was no ICU level care. There were no mechanical vents. And so they really had to focus on non-pharmacological means, and definitely no vaccines. So what did that look like? So there was definitely a lot of isolation, a lot of quarantine, wearing masks. Schools were actually closed down. They utilized disinfectants, and they limited public gatherings. And it really wasn't until 1997 that researchers were able to actually use the virus and kind of sequence it to determine and actually how to study it. So they determined that there were specific genes that increased mortality, increased weight loss, increased the rapid replication of the virus, and specifically lung damage. And so that was kind of what we learned about, or what we learned from the Spanish flu. And Brenda, I'll let you jump in and kind of discuss H1N1, which is its predecessor, or what comes after.
Sure. Thank you, Michelle. Of course, we cannot talk about pandemics without talking about the swine flu that came around in 2009. That is probably the one that probably comes to mind for most of us millennials. Of course, this, like I said, it was first, I guess, seen in Mexico in 2009, and this was actually the first event that actually it prompted the activation of what was known as the 2005 International Health Regulations by the World Health Organization. This basically, it delineated the responsibilities of each country when it came to something like a pandemic and the role of the World Health Organization. So this is kind of where we got a lot of the reporting requirements, as well as some of the trade and travel restrictions that we have seen with COVID. Kind of going off of that, a lot of the actions that were shaped, a lot of the actions that kind of helped guide the world's response to H1N1 was actually linked to the SARS outbreak in 2003. This is kind of where, SARS was first seen, I believe, in late March, and by July of the next year, it was basically gone. And a lot of that had to kind of do with a lot of the collaboration that had to happen in order for the outbreak to be really quarantined and contained really fast. I think with our current the current pandemic now, there's just been a lot of collaboration that has happened, and I think the biggest testament to that for us has really been the development of the COVID-19 vaccine. The COVID virus, the current one versus the SARS virus, they're very similar. I believe they share about 80% of the same 80% of the same genome sequence. So that's just to say that we weren't really starting from scratch. And so when people ask how were we able to really develop a vaccine so fast when we didn't have one for MERS or SARS, just we weren't really starting from scratch.
Exactly. So MERS was another, Brenda mentioned MERS, which is the Middle East respiratory syndrome, and that was discovered in 2012 in Saudi Arabia. And so it was kind of isolated to the Arabian Peninsula, and actually only about 2 cases were found in the US in 2014, both related to patients that actually traveled, worked, and lived in Saudi Arabia. And so while there weren't a lot of deaths associated, about 858, the percentage was really high. So about 35% of patients actually passed. So the total number of cases was only like about 2,000. But interestingly enough, and we'll get to vaccine development really quick or pretty soon, there was an outbreak in 2015 with MERS in Korea. And a lot of the lessons that were learned from that outbreak were more about processes in the hospital, I would say. And so they really understood that, you know, the outbreak was driven by super spreaders. So I thought that was a word that started to come up during COVID times, but this is definitely pre-COVID. They also kind of put stipulations on visitor control or having visitors in the hospital, which is what we see now. They started training healthcare workers regarding different infectious diseases, strengthening the healthcare infrastructure, and ultimately preventing, kind of shifting the thought from prevention, from treatment to prevention. And so with MERS and SARS, essentially there were phase 1 trials for these vaccines. So I think a lot of times, like Brenda was alluding to, people think that this COVID vaccine, you know, was created super quick, but they did have some stepping stones. And so the SARS and MERS vaccines did have phase 1 trials, and they'll talk about this in a little bit. But due to the limited resources in terms of funding and the number of cases, there weren't that many cases, as we discussed. There wasn't really a lot of interest in pursuing phase 2 and phase 3 trials. So I'll go ahead and turn it over to Yewande, who will start to discuss some of the vaccine development or production.
Awesome. Thanks a lot, Michelle and Brenda, for that awesome discussion. So I'm just going to go real quick and talk about what typically does vaccine approval, what does that process kind of look like in a normal world, a non-pandemic situation? So usually you have a drug company that is interested in pursuing vaccines for a specific virus. There usually are viruses, and they start by working more in the lab or like in making simulations based off of information that we know about the virus. That usually can take some time to kind of synthesize the perfect molecule or the perfect candidate that they think will work. The next step of that is usually then going into something that we call preclinical testing. So they test in animals. Usually a lot of times they'll use mice. They'll test in mice to kind of test out the hypotheses that they have. Does this work? Is it safe? What does it kind of, what are some issues we can anticipate in humans? Based on the preclinical aspect or the preclinical data, they then go on and submit an application essentially to the FDA saying, hey, we have this candidate, we want to start testing this in humans. Before they can actually start testing it in humans, they have to get approval from the FDA. The FDA okays that, and then they do the first stage, right? And so they test it in humans, first to make sure that it is safe in humans. So they do a phase 1 with a very small number of people, about 100 people or less, to test mostly for safety in healthy people. Then they do a phase 2. The phase 2 usually gives a little bit of safety information, also gives us some efficacy information in terms of dosing. So that's how they will determine the appropriate doses to use. And then they go on into a phase 3. After they've done their phase 3 study, they then turn in another application to the FDA. The FDA reviews this, and in the FDA's review, they're reviewing for safety, they're reviewing for efficacy, they're looking at manufacturing practices. It's a very, 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 very detailed review that essentially looks at every aspect of this vaccine, from production to administration, to make sure that this is something that we should roll out to humans. After the FDA has reviewed it, if they agree to move forward with it, this goes on to company, and they can actually go on and start making the vaccine for distribution. There's an added step that usually is involved, especially for vaccines that potentially we need to have on a schedule. That's where the ACIP, which is essentially a body of independent experts that come together to review all the same data that the FDA has reviewed and make recommendations for who to give this to, when to give it to them, and all of that. So that was a similar process that we saw with both the Pfizer and the Moderna vaccine, where all these different groups that are all independent from the FDA essentially advise the FDA, but also advise the CDC and advise all of our medical professionals on how to use these vaccines. After these vaccines are rolled out, the next step is something called post-marketing survey. And this is a typical process, not just for vaccines, but honestly, for every single drug that is on the market. And what is a post-marketing survey? It's essentially a system where the drug company, the FDA, the CDC, depending on the specific indication, are all reviewing information based on safety, especially for these vaccines that are rolled out, to make sure that they are continuing to be safe for people. So all of the adverse reactions that people may have, any weird interactions that may come up after we started giving it to people, that we find out usually in this process. How does this then tie into a pandemic situation? So in a pandemic situation where right now we're at a stage where we have 3,000 Americans dying every day from this vaccine, we cannot wait 15 years to get a vaccine. And so that's where the FDA has, as part of their tools that they have in their toolbox, these mechanisms where we can fast track things without skipping the safety pieces of it. A lot of what gets cut out is the bureaucracy. The FDA reviews that need to happen, they have them essentially at the same time as the studies are happening. So it's a rolling review. So the FDA has had access to the data for the COVID vaccines since about May, when the studies kind of started, and have been reviewing all of these things. And the other aspect of this for the US was the funding. And that has always been an issue with vaccines, is who's going to fund them, who's going to invest in them. What the US government did was they promised all these companies that they will take on the financial risk of pushing these vaccines and doing the studies on them. So the biggest thing, or the biggest space that the funding happened in, was in Congress allocating about $10 billion specifically for vaccines, and also for other therapeutics. With about $6 billion of that went to vaccines. And what that did was the US government essentially paid all these companies to start making the vaccine before we even knew that they were going to work. And the reason why that was important is that usually after the FDA has approved the vaccine, there's usually a lag time in when the company then has to upscale for mass production. So the US government essentially took that risk on saying, we don't know if this works yet, but we're gonna pay for this product on the hope that it will work when we get all the safety data. And so that was a huge part of it. The other aspect of it was this was one of the biggest global collaborations that we had in terms of scientists and clinicians around the world, where once the Chinese saw the virus first, they were able to sequence the virus. And once they were able to sequence the virus back in January, all of these companies were essentially able to see what they needed to essentially target to make a vaccine. The other aspect of it was a collaboration with so many different agencies within the US. So you have the HHS, the CDC, the Department of Defense, the FDA, a lot of academic centers, a lot of private companies in terms of the big pharma, and also across the world. So we had studies running in the US, we had studies running in the UK, we had studies running in Brazil and in so many other countries. And everybody essentially stopped and focused on trying to get a vaccine. And so that's what this process kind of looked like in terms of comparing it to what typically vaccine development looks like. And I'm going to turn it over to Robbie to talk a little bit more about what the vaccine technology looks like and what the EUA, the FDA approval process kind of was like for this vaccine. Thank you.
Awesome. Thanks, Yewande. Perfect. So yeah, like you said, the emergency use authorization is the EUA that we call it. I keep calling it EAU, so bear with me. But it's not a new process at all. There's a section in the Federal Food, Drug, and Cosmetic Act where it's section 564. If you want to look it up, I always look up things that I don't know about. So if you want to look it up, it's section 564. But that's letting us know that the FDA commissioner may allow an unimproved medical product or a product that has been approved, but for something else, and we want to use it for a new indication, it may be approved in the case of emergency. So an emergency, like there's an emergency situation going on called COVID-19, we all know about. On February 4th, 2020, actually is when the Secretary of the Department of Health and Human Services did declare COVID a public health emergency. At that time, now we knew we could move quickly to try to get this vaccine or anything dealing with COVID to get it under control with diagnosis, treatment, or prevention. So that's where we are now, and how this EUA works is a company already has went through these phases. They went through the research and development she talked about. They went through the preclinical trials. They started their phase 1 and 2 and 3 trials, and they have a lot of the data that they will use for their full application, but they write a letter called the EUA to the FDA and other federal agencies asking if their product could be used early because of the state that we are in of that emergency. The FDA does review that paperwork, they review the application for scientific data, everything that the company or industry manufacturer has, it is reviewed by the FDA. No steps are skipped, all steps are followed, and it's really an open, very transparent process. You can go to the FDA website and look at the letter that was submitted from Pfizer. You can go look at the letter that was submitted from Moderna and see all of the scientific data that was there. Also, you can see the response that the FDA had. So there's no secrets when it comes to this is a product that is not FDA approved yet, but is working on that process. And I think it really is a good process. There are currently 312 EUAs for COVID-related things with diagnostics, treatments, and for preventative measures. So they're really working hard, but not everything that is submitted is approved because not everything meets criteria. So they, like I said, they look at the totality of the available data, make sure the evidence is clear, and they make sure that they state that this data will support that the known potential benefits, right? The benefits that are known will outweigh the potential risk. Everything we have in science, there in life, really, there's risk and benefits. So the FDA is saying by approving an EUA that they believe with the data that was given to them for people of the world that we need to treat, the benefits outweigh the risk. So I want you to always think about that. There's never a 0% risk. We would never have anything if we always had to have 100% guarantee or 0% risk. So the FDA did do that with both the Moderna and Pfizer vaccine, Pfizer BioNTech vaccines currently. So the quality, the effectiveness, and the safety has been checked out, that it will, they checked out that information to make sure that it was available for the people who wanted to look at it, also that it was going to be beneficial. And the experts, the FDA experts say that even though you have EUA, you still have to go through the processes to continue to apply for the licensure to actually get FDA approval. For instance, for the EUA, if you remember when we were deep down in COVID and we had no drugs, we had nothing, and we were talking about hydroxychloroquine, and which is the anti-malarial, and using that as a potential option because we were at a desperate need and nothing else. We had things in the pipeline, but nothing that was created that my dog is right here by me, but we had things in the pipeline, but nothing that was valid that we could use right now. So anti-malarials have been used in the past, that there shows that there could be some slight data that could be beneficial. So the EUA was approved in March 2020. But after it being used in a few patients and we saw that there were side effects and we saw that, this wasn't the right option and there were things that were being produced with other EUAs, and there were cardiac risk and adverse effects, the FDA stopped it. It was removed, it was revoked, and that was all public. And if you go to the FDA, you can see all of this that I'm discussing and talking about here. So for safety, if there is a safety concern, things will be removed if any safety concerns are seen. So that helped me with my decision to get the vaccine on the 4th as well. And Yewande, she talked a lot about Operation Warp Speed when she talked about the partnerships and the Health and Human Services, how the CDC is involved, how everyone has one goal. And if we get everyone together with one goal, of course, things will get done. And I think that's why things have got done really fast is what we think, but really it's right on time. We needed this to happen like yesterday, which it happened few weeks ago now. But we needed things to move this fast because COVID has taken our lives very fast as well. When it comes to funding, she talked about billions of dollars being given to these companies so they don't have to worry about working on other things. First of all, like they can focus on COVID vaccines. So this makes sense that it's getting done quickly because they're all able to focus in. We have the best scientists, the best doctors, the best industry manufacturers, like the best defense. We even have military helping us with the logistics of the operation itself. So when you have funding and you have the right people, that speed is warranted. Like we need that speed. And I think a lot of that has to do with the simultaneous manufacturing of the vaccine. They manufacture several types. And then when they find out like this is the one that works, then they continue manufacturing that one or distributing once we know which one that really is. But there is a lot of money lost if it was just a company. But when you have the government footing the bill, it makes sense that things were happening like with speed. So thank you for sharing that section with me, Yewande. I also want to say with all the data, I'm sure you guys are going to look things up. CO, I definitely would, keep looking things up. But mRNA technology is not novel, it's not new. It was first developed in the 1990s, like around the time I was born. But it was found not to be real stable because of the technology that we had then. There are some trials that already use mRNA with HIV, rabies, Zika, even influenza. We have some clinical trials that are ongoing with the mRNA technology. So while all of this may seem like it's happening overnight and really quickly out of nowhere, that's not true. But I do, however, understand people being skeptical, right? Like Black and Brown communities have mistrust, rightfully so. Like there's institutional trustworthiness that can be an issue, not because of us, but because of what has been done to us. And I like this saying, like, fool me once, shame on you, fool me twice, shame on me. So a lot of us are protecting ourselves. We're like, I'm not getting anything I don't trust. I'm not putting anything in my body I don't trust, but y'all be eating McDonald's though, and y'all don't know what's in it. But I think a lot of it goes to like, I can't say no, because we can go back to talk about the Black community and how slavery, we were property and we couldn't say no. There was no saying no. We listened to what was said or our ancestors, because I would not have made it. But now I think we are like, okay, we can say no. And that's why I'm saying no to this. Like, I'm not giving it a chance. I don't trust it because all of these things are going on. We can talk about Henrietta Lacks. We can talk about how her cells were removed from her body and they're used to this day without getting her permission, also without giving her family any compensation, and still science is going on without doing that. We can talk about more recent events. We can talk about the Tuskegee experiment from 1932 through the 70s. Like that's close. Like that's the time where grandparents were born, and which have brought a discussion to many of your reasons for you not wanting to obtain this vaccine, that Tuskegee experiment. So I'll let us dig a little bit deeper here. I want to let you all know that I hear you when you say you don't trust, but please hear us when we talk about how this will never ever happen again with the structures that we have set.
Excellent segue, Robbie. So I know there is a lot of hesitation when it comes to trusting what the government is putting forward for the Black and Brown communities. And it really does span or, come from a lot of different things, including the Tuskegee experiment. So before I talk about what happened in 1932, what actually was the start of the experiment, I want to go back to 1927 when syphilis really became a major issue, right? A major public issue, and they needed treatments. So just as we discussed vaccine development, they also needed to develop drugs to treat syphilis. And so a few years later, they tried mercury, they tried bismuth, and other, treatment options. But again, as Robbie mentioned, in terms of risk versus benefit, they were having low cure rates and really high side effects and mortality associated with those treatment options. And so around 1930s is also when the Great Depression was, starting, and funding started to be cut in a lot of different projects, including this type of study. And so the purpose was that they wanted to continue to evaluate non-treated syphilis patients in order to see how syphilis took over the body. And this was all to kind of support their need for additional funding to continue to find an actual treatment. That was the initial intended use. But what we learned is that over time, syphilis actually, so there was actually 600 men, Black men in Tuskegee, and I'll look at my notes, 399 actually had syphilis and 201 patients did not. And so they told the patients that they would receive free burial services, they would receive free medical care, free food. And at that time, there weren't a lot of things that were going well for a lot of people during the Great Depression. So what do? You say yes to free medical care, you say yes to, free meals, and they were under the impression that they were going to be treated as well. What they were not told is that the purpose of the study. So they were not told that they were not going to be treated. They were not told, they did not give informed consent, meaning that they only agreed to just the exams and treatment. And so a few years later, so this all started in 1932. In 1936, there was actually a study that was published that kind of highlighted the unethical concerns of this study. And it was kind of like brushed under the rug. They continued the study. 1945, penicillin was finally determined as the drug of choice, which we still use today. In fact, when patients come into the hospital with syphilis, I quickly look to see if they have a penicillin allergy, because that's what I'm giving 100% of the time. If they do have a penicillin allergy, I'm gonna give them the desensitization so that they can get the penicillin, because that still is the #1 choice for syphilis. But right after syphilis, or right after penicillin was determined the drug of choice, they still did not give the patients that actually had syphilis treatment. So that was a huge flaw in the project. And then as the years passed, a study was actually published by the Associated Press, and that kind of further highlighted that this study had been going on for 40 years now, and that patients weren't receiving treatment. And so this is something that is really, it really affects the Black community, because it comes from we create a lot of mistrust, or that has created a lot of mistrust in our community. Even though this wasn't even a vaccine, this was supposed to be a treatment study. But this, kind of parallels and definitely has some historical implications. So I'll touch on a few of them, and I'll let Brenda address some of the other historical implications. But we don't want to be experimented on. We don't want to be the ones that, get the vaccine and there's only been 40,000 other people. We are hesitant, even though there were 9% Black people in one study and 10% in the other study. We're still pretty hesitant when it comes to this all of a sudden new COVID vaccine, and we don't want to be used for research. We have, a whole experiment that lasted 40 years that has shown us that we shouldn't, we should be cautious when it comes to being used for research. And even still in the 1990s, there was a study that I was reading about where they specifically focused on Black boys, and these Black boys, and they wanted to link genetically a cause to aggressive behavior, and they only sought out Black boys. So this is still something that is going on, and we don't want to make light of the situation, but we definitely, you know, want to present the facts so that you guys can make an educated decision. And I'll let Brenda discuss a little more about some of the other historical implications in the research guidelines.
Sure. Michelle, I think you pretty much touched on just about the most important parts of it, but I would just like to echo that definitely the Tuskegee study, it definitely laid the foundation for African Americans having this continued distrust of the medical establishment. And this does carry on when we do talk about vaccines as well. There's evidence even past the 1970s when this particular study was done. There's evidence that, you know, when we had the AIDS pandemic, there was still a lot of hesitancy in our communities when we talk about going to a doctor. There is the belief that, you know, HIV and AIDS is a form of genocide. You have people thinking, you know, the U.S. government is, you know, promoting drug use in black and brown people. There's the belief that HIV is a man-made weapon of racial warfare. But I think it's also important for us to know that in modern times, it would be very, very hard for somebody to even try to remotely replicate what was the Tuskegee study. There has been a lot of steps that have actually been taken to prevent these exact same things from actually happening. So right after that, I guess when the results of the Tuskegee study were finally brought to light, the CDC, the Department of Health and Human Services, they actually put together a law that basically would have a lot of the would have a lot of the protections that were missing from this particular study. So we have now informed consent. Before a person is put into a study, they have to know what is it for, what am I being studied? Is there anything that you have to tell me that may make me not want to actually be part of this study? There's independent review boards. There's we have the FDA, and in a lot of studies, both for the Pfizer as well as the Moderna, there were independent review boards that looked at the data, not just the people making the actual vaccines themselves. So that's really all just going to say that currently there's so much there that has been put into place in order for something like Tuskegee not to really be able to happen again. And so with that, I will let Jacinda kind of just touch on the actual vaccine itself, the technology, and just some of the data behind it.
Hello, everyone. It's me again. So I'm here to talk about the COVID-19 vaccine itself, which from what I can see in the chat is just the topic of discussion. So it is my hope that I have prepared well enough so that I can address most of these questions, and the ones that are left behind, we'll make sure to get to before we wrap up the presentation. We are also prepared to go beyond the hour-and-a-half mark. So with that being said, if you guys, if we hit that hour-and-a-half mark and you see that your question hasn't been answered, rest assured, we will be sure to get to it. So let's just do a brief review on vaccines, period. So we've done a lot of discussion about, you know, previous pandemics, how they prepared us, the vaccine production process, the Tuskegee experiment, and how that institutional distrust may have lodged just a certain amount of fear of us in getting those vaccines. So let's talk about vaccines. So you can have different types of vaccines. So you can have what's called a live vaccine. So what's the live vaccine? So it's a little bit of the virus a virus or a bacteria that's injected into you, and then your body mounts an immune response. That immune response looks like what we know as the development of antibodies. That's what antibodies are, our body's immune response to the introduction of a virus or a bacteria. So that a live virus is an actual small part of that actual of that virus or bacteria. It's a small part, small enough to where your body can mount that immune response, but not actually suffer the events of the infection itself. You can also have an inactivated vaccine. So an example of an inactivated vaccine would be something like a hepatitis vaccine, something along those lines. So what is an inactivated vaccine? What that is a killed part of a virus or a bacteria. So your body sees that killed part or that dead part of that virus or bacteria, and it once again mounts that immune response and develops those antibodies. Then you can have, we have another type of vaccine, and that's when your body, you are injected with a specific part, a specific subunit, or some type of significant part of a virus, some distinguishing factor of a virus or a bacteria. And then your body mounts an immune response, develops antibodies. So should you ever come into contact with that certain bacteria or virus, your body will be able to protect you from against that. An example of that would be like the pneumococcal vaccine. So kind of thinking about this brief overview that I've gave you guys about vaccines, the COVID-19 vaccines that are currently available, they use a pretty significant and just, I am a scientist. So as I told you guys, I am a bench scientist. That means that I love this type of stuff. This is what keeps me up at night. So they use a sophisticated technology. They use mRNA technology. So what's mRNA? I know we see this thrown around. And then my favorite question is, is this a chip that will be injected into me? Honestly, I wish it were. It is not. It is not that exciting. So mRNA is commonly available in our bodies, right? It's something that, so mRNA actually allows for, it's called messenger RNA. So it sends messages to our cells on actions that our cells should carry out. So this vaccine kind of follows that same process. So in the COVID-19, in the coronavirus has a very, very specific type of makeup. It has what we call the spike protein. Viruses are genius. They are smart. They know that each virus has a very distinct kind of factor that makes it more apt to cause infection in individuals. With the coronavirus, what makes it so active, what gives it just that extra kick is the fact that it has these spike proteins. So what do these spike proteins do? Let's think about if you were to step on something with some spikes, you would be able to open that cell up. So that's what this spike protein does for the coronavirus. It pokes that cell, and then it allows for that virus to just infiltrate that cell. And then we see what it is, what we know to be true of the coronavirus. We then see that respiratory depression and everything that ends up happening once you are infected with the virus. So this mRNA technology, which I told you guys is novel, it's amazing technology. What it does is it delivers your cells basically an instruction manual. So let's think of this as IKEA. So it then takes the instruction manuals to your cells, and it says, hey, here is what this spike protein looks like. You need to go ahead and you need to make this specific spike protein. So remember, I told you guys about live vaccines. I told you about inactivated vaccines. And I told you that once our body sees this, it then mounts an immune response. So once our bodies get that message from the mRNA to make that spike protein, the mRNA is disposed of. The mRNA has done its job. It is not in any way integrated into our DNA. Its job is done. Our bodies then go ahead and it mounts its immune response. Meaning that if you were ever in real life to encounter the coronavirus, your body would have seen the coronavirus. It would have made it itself through that injection of the mRNA, and then your body would have mounted an immune response. So I got the question, which I thought was really interesting and honestly a good question. I got the question of, do Black people have less spike proteins than other people? So if they have less spike proteins, then of course that would make sense that they would be, then the vaccine wouldn't work. So about these spike proteins, as I told you guys, viruses are smart. So they have very distinguishing factors to make them cause the infections that they do, to make them what we call in the scientific world, to make them more virulent, to make them so that when they get you, they get you. And this is a distinct factor of the coronavirus, the spike protein. So everyone, whether they are Black, whether they are Asian, whether they are Latinx, whether they are white, if they have the coronavirus, then they indeedingly had a spike protein that went ahead and was characterized and was signature of that coronavirus that they did have. So I thought that was a good question, but I wanted to make sure that I elaborated on that. So then I saw in the chat, which in my head, I knew this question would come up. Why is it that we have to get 2 doses of this vaccine? Once again, phenomenal question. So let's think of this as, and you know, I'm a child, I have a parent, and my parent is online here in our discussion. And let's think of our bodies. Let's think of this vaccine as a parent. So as a parent, you tell your kids, clean up your room. So let's think of the vaccine, let's think of the first dose of the vaccine as your parent saying, clean up your room. It's that first list of instructions. So that mRNA is injected, you have that first set of instructions. You need to make this spike protein, you need to develop these antibodies. But of course, you know, like kids, our bodies, they get the message, but they don't get it loud and clear. So that second dose is, hey, I told you, you need to clean your room. So that second dose is, hey, this is this spike protein. You need to make this. You need to make the antibodies against this specific spike protein. So that is why we need 2 doses, but also Yewande and Robbie did a phenomenal job. Dr. Dio and Dr. Christian did a phenomenal job at really talking about how we go through these different phases of study construction. So as they matriculated through these different phases and moved through these different phases of study construction, what did they see? They saw that those individuals, they were protected after they received dose 1, and we'll get more into the study discussion later, but they were protected after they received dose 1. But when they received dose 2, they were very protected. They were confident after they received that second dose that they would, in fact, in those groups that had received the vaccine, they would be less apt to actually succumb to that COVID-19 virus. So with that being said, that is why we now do 2 doses of the COVID-19 vaccine rather than 1. However, as Dr. Christian mentioned, there are other studies underway. There are other vaccines, you know, in other vaccine development underway. I know I love Dr. Fauci. I listen to him often, and he often talks about the Janssen or the Johnson & Johnson vaccine. So they are more proactive. Well, we won't say more proactive, but they're very proactive in their study procedures. And they're currently trying to see, can we give 1 dose or can we give 2 doses? Do you have that same type of efficacy? Do you have a similar amount of patients that actually succumb to COVID-19? Should they receive 1 dose or 2 doses? So there are studies underway, but as of right now, and with the vaccines that we currently do have available, you do have to receive 2 doses. And as I mentioned, why is that? That is really that is definitely our bodies and our body's immune systems are definitely the case as to why we have to do that. We have to make sure that our bodies have received that message loud and clear on how to go ahead and make those antibodies to that specific spike protein or the coronavirus that was injected into them. So then I also saw in the chat, and a question that I've gotten just from my family members is, if there are resistant strains, why is it that we get this COVID-19 vaccine? Is this COVID-19 vaccine still going to be, you know, something that works? So, you know, this is great news for you all, I guess, and better news for me. So I'm a resistance researcher. That's what I do. That's what I've dedicated my, you know, life's work, my young life's work to, is researching resistance. So what is resistance? Let's really define what that is. So bacteria and viruses like COVID-19, they are smart. And it's something that we often talk about in the lab, in the research world as well. You know, these viruses are very smart, intelligent. When they can sense that your body is able to form specific responses to them, meaning that they can sense that, you know, at some point, this person's body is going to be used to me. they're gonna be used to seeing what it is that I can do. So I have to change in some way so that their body's not able to form an immune response, or the antibodies that they have aren't able to protect them against what it is I look like. So that's essentially what resistance is. So resistance is when the original strain changes in various parts. So you can have multiple changes, you can have one change, things of that sort. So I think that the big thing here with COVID-19 is to kind of think about it. And I saw the question is resistance similar to the flu vaccine with COVID-19? It is not. So with the flu with the flu virus, so the flu virus rapidly changes. And that's why when you get a flu vaccine, they'll say, oh, there are several strains of the flu inside of this specific vaccine. So what that means is there are several flus, or so say that there's an original flu virus. That original flu virus has changed multiple times. So we have combined all of that original flu virus plus those changes into one vaccine. And you will take that with the hope that we have captured all of the changes that you would experience out in the world, and your body will form antibodies against it. So should you come into contact with those strains that we have protected you against, you should be fine. So that's the idea with the flu. The coronavirus, and in all honesty, this is the only thing that the coronavirus has given us. It does not mutate the way that the flu virus does. So there are changes in it, absolutely. Did they really observe resistant strains in the UK? Yes, they did. Are those resistant strains probably in the United States? They possibly are. I will not be, they are here, yes.
Colorado.
I will not be, I will be very transparent in that. However, rest assured that when your body develops those antibodies or develops that protection, see here how I have this picture of the spike protein. It does not develop protection against one part of that coronavirus or against. So how we see these different spike proteins and we see these different parts of the coronavirus, our bodies are able to form antibodies that can attach to different areas there. So you have different types of antibodies. So should there be one change, that won't completely knock out the vaccine. And why is that? It's because it's allowed for us to develop different types of antibodies that could work in different ways to protect us. So I'm not going to minimize resistance because, like I said, if resistance wasn't something that readily occurred, if resistance wasn't important, then I would not have dedicated my life's work to researching it. So it is important, but at this point, do we feel that it decreases the efficacy of the vaccine? No, we do not. Do we feel that you are protected with the vaccine against these possible resistant strains? Yes, we do. Has this been researched by the scientists that have developed the vaccine? Yes, it has. Dr. Kizzy and her lab, they, like I said, scientists on the bench, we know that resistance occurs. Therefore, we work overtime to combat this. So Dr. Kizzy and her lab did come across strains that had changes, meaning that they were resistant, or meaning that they were mutated, and they were mutations of that original strain. However, when they tested those strains against the coronavirus vaccine that they had developed, those strains did respond to that vaccine. So with that being said, and I hope that I have spent enough time on resistance. However, I'm sure there will still be questions and I will be sure to answer those again. But as of right now, that vaccine will work against it. So this is when we run into that problem of, will we need to get another vaccine to protect us? Will this resistance become a big deal? Will at some point this coronavirus get smart enough to mutate in multiple places, rendering the vaccine ineffective? This is possible. I will not, minimize that risk. I will not downgrade that possibility. Is that our current possibility today? No. Will it be our current possibility a year from now? Possibly not. However, as much as the research has taken place into these vaccines, please know that there is research taking place into how to modify these vaccines to ensure that we can be protected should we see it again. So, or should those mutations continue to appear? Once, talk about that as well. This is where we see that importance of ceasing travel. This is where we see that importance of wearing masks. And that is because when you go to other areas, you are bringing what it is that you have in your area with you. So the interesting thing is that that strain from the UK, that resistant strain, guess where it came from? Not the UK, it came from South Africa. And why is that? That's because people did not adhere to the travel restriction. So people travel to the UK. So when you travel places, you have your own bacteria there. You have your own specific coronavirus strain in that place that you are in. So when you go to other places, you then take whatever it is that you bring with you, you take it there. So you don't go home, so you don't leave with nothing. So always remember that is why those travel restrictions and that mask wearing, that's where that importance comes in. So, I also got other questions about immunity. And, how do we develop that immunity? And how does that development of that immunity of the immunity look like? So it looks like, so we develop that immunity, and immunity doesn't come without its own collateral effects. So the development of immunity looks like when we have just that, when you have a fever, or when you have a headache. So why is that? That's because, as I mentioned, your body is seeing a small amount of this virus, of which it is to form antibodies against, or to form protection against. So it's not seeing enough to where you would actually experience, the serious events that you would experience should you see, that vaccine, that virus itself. However, because your body is, seeing this foreign invasion of something that it knows it's not supposed to be there, you'll have a fever. And that's just our body's natural response to an invader. Our body's natural response is to mount a fever, or to increase our body's temperature to get that virus or to get that bacteria out of the way. So that's why you will have that fever, that is discussed in that in the COVID-19 trials, or when we talk about COVID-19 and just the effects that you can have there. However, with that fever development, I think that it's important that we mitigate just that fear, and we say, bravo to your immune system if you do get that fever, because you know that should you ever come into contact with that actual COVID-19 virus, that you are protected, because your body did in fact develop those antibodies, did in fact develop that immune response to COVID-19, and you have the fever to show for it. We will move into the clinical trials in just a bit, but I think that it's also important to note that in those individuals that did, have a fever, and I will say this probably 3 or 4 times, because I just want to reiterate this. They did, take Tylenol, and they did take ibuprofen, and that fever did resolve within 1 to 2 days, which is expected. So then, as we are talking about this, building immunity, as we're talking about the vaccine itself, as we talk about resistance, I think it's important we talk about efficacy, right? So what did the clinical trials say? What does that information mean? And what's our takeaway from that? So there were clinical trials that were conducted, of course. So we had the Pfizer trial, and then we had the Moderna trial. And then I'll go through both of them. And this, as Dr. Dio kind of explained to us that process, you have the phase 1 trial, you have the phase 3 trial. So this is what we know as phase 3 trials. So what do phase 3 trials entail? They entail a vaccination group. So participants that actually receive the vaccine, and then they contain a placebo group, or individuals that receive absolutely nothing, nada. So these trials are randomized. What does it mean to be randomized? It means that the individuals that partake in the trials, as well as those individuals that are conducting the studies, do not know who it is that's receiving the vaccine and who it is that is receiving nothing. And why do we do that? To remove any biases. So you wouldn't want for more people to receive the vaccine than for more people to be in the placebo group. You want to be able to kind of like make sure that it's equal across both groups. So that is why we do have these phase 3 trials, and that is how they are conducted. So let's get into the Pfizer trial, because personally, I spent a lot of time with this trial, probably too much. But in the scientific world and the ID world, and I'm sure that my sisters can attest to this, we were so excited when it came out. I think Michelle sent several text messages, "Hey guys, here we are." So yeah, there were about 40,000 participants in this trial total. So with those 40,000 participants, there were 20,000 individuals that were in that actual group that received the vaccine, and there were 20,000 individuals in that group that did not receive the vaccine. So 20,000 that received absolutely nothing. Why does this make sense? Why would they do this? Because they wanted to see, is there a difference between if a person were to get a vaccine or if the person were to get absolutely nothing? Does this vaccine have any type of efficacy, in comparison to absolutely nothing? So as I said, there were about 20,000 individuals in each group. And then I think it's important to talk about the numbers, since we are here talking about efficacy of those vaccines. It's important for us to talk about the Black participants in each group. So in each group, there are about, so in total for the entire study, there was about 3,400 Black people that participated. And in that, of that number, there are about 1,700 Black people that were in the placebo group, so that received nothing. And there are about 1,700 Black people that were in that actual vaccine group that actually received the vaccine. So I just wanted to make sure that we had that in our minds as we continue to move through this information. So what does that mean? As Dr. Maxim said, that means that Black people represented about 9% of the individuals included in this trial. So what does this not look like? Tuskegee, right? So we are in the minority. This does not look like the Tuskegee study. So with that being said, what did they see in this trial? So what did they see when they after dose 1? So after dose 1, so days 1 through day 12, they saw a decline in those individuals that were actually diagnosed with COVID-19 in that group that received the vaccine when compared to those individuals that did not. So in that group that actually received the vaccine, so prior to day 12 of receiving that second dose, there were only 39 individuals that were infected with or diagnosed with COVID-19. In the group that did not receive the vaccine, there were 82 individuals that were diagnosed with COVID-19. So what does that let us know? That means that prior to day 12, prior to that second dose anyway, there were 52, it was a 52% protective rate there, right? So 52% protection. So while you are protected from COVID-19 after that first dose, let's move into what they actually saw after that second dose. So after that second dose of the vaccine, that's where it really gets interesting. So then after the second dose, they only saw 8 individuals in that vaccination group that were infected with the COVID-19 virus. So I don't know if anybody else feels this way, but I think that is just absolutely spectacular. So of those individuals that tested positive for COVID-19 in that vaccination group, there were 8. So in those individuals that did not receive the vaccine, there were much more than 162 individuals that did develop the COVID-19 virus. So what does that mean percentage-wise? That means that the vaccine was 95% effective. How amazing is that? I cannot tell you the joy that fills me with, because I think that our most effective vaccine to date is the measles, mumps, rubella vaccine. And I can't wait to tell you guys how effective this vaccine is. It is 98% effective. So what does that mean? That means that we are 3% off. In a study that included more participants than any study or in between Moderna and Pfizer, more participants than we have ever had for any vaccination trial. How amazing is that we saw that efficacy there? So I think that's great. But of course, I would be remiss if I did not talk about, events or adverse events that occurred in these groups. And as I said, I believe in a spirit of transparency and a spirit of honesty. So there were about 52% of individuals that described having any type of effect after receiving this vaccine. So what does that mean? That means that they could have had a localized reaction, meaning that they may have had pain in their arm following injection. I received the vaccine, and I will say that I did have pain in my arm following the injection. There were also individuals that reported having a headache. There were individuals that reported having diarrhea. There were individuals that reported having a fever or having chills. Those individuals that did report having the fever and the chills did say that after they took ibuprofen or Tylenol, that fever did subside within 1 to 2 days. But as I mentioned, those effects are common. The pain at the injection site. I also have pain at the injection site when I get a flu vaccine. That's just from the formulation of that vaccine being injected into your arm. And then as far as the fever and the chills, as I mentioned, that is your body's way of mounting that immune response against that virus that is being introduced to it. So let's get a bit into the Moderna trial. I just want to talk about what we ended up seeing there, and pretty much the similarities that we noted between Moderna and the Pfizer trial. So Moderna, once again, just beautifully constructed, in my opinion, as far as the scientific process goes, as far as just how they went ahead and developed this study. So there were about 30,000, a bit over 30,000 participants in this trial. This is not publicly, well, it is publicly available in the FDA document, but they have not published the manuscript of their findings as of yet. But so as I mentioned, there were 15,000 participants that received the vaccine, and there were 15,000 participants that received nothing. But with the Pfizer study, I want to go into how many of those participants were Black. So there were 10% participants that were Black in this trial. But I also want to give special attention to Moderna. While Dr. Kizzy was on the front lines of this Moderna vaccination development, so I am already biased because I am a supporter of Black women, especially Black women in science. However, one thing that I would like to attest to is that, and Dr. Christian made sure to inform me of this, is that Moderna actually stopped their study because to the current date, prior to them stopping their study, they only have 4% of Black participants enrolled, likely for the reasons that we mentioned, institutional distrust, right? The historical implications of the Tuskegee experiment that kind of led us to that distrust. So, and as we all said, understandably so. However, they knew that because Black participants were majorly affected by this virus, it was important that we knew how this vaccine would perform in them. So there were 10% of Black participants in this study. While that looks to be a small number, it is quite similar to the 12.5% that Black people represent in the United States. Additionally, that 10% is the third most populous racial and ethnic group that was included in that trial. So while it looks small, it is quite representative. So what did they see after dose 1? So this is prior to 14 days. They saw that there were 7 individuals in that vaccine group that had been diagnosed with COVID-19 versus 39 in that placebo group. Then as they moved into 14 days, prior to that second dose, so they moved a bit further out, they saw that there were only 2 people diagnosed with COVID-19 versus 28 in the placebo group. So see how we are kind of just moving towards just, while you might be protected, against COVID-19 after that first dose, first and foremost, it takes our body a while to forge just that immune response, for one. And then for 2, we're moving into that, what does it look like closer to that second dose? So following that second dose, there were 11 participants in that in the group that actually received the COVID-19 vaccine that were actually diagnosed with COVID-19 versus 185 in the placebo group that received nothing that were diagnosed. So what does this look like in terms of efficacy? There was 94.1% efficacy. Once again, I am jumping for joy because how amazing is this? As I stated, the most effective vaccine that we currently have to date is the measles, mumps, rubella vaccine, which is 93% effective. So, of course, once again, I would be remiss if I did not bring up the fact that there were about 80% of individuals that did, discuss that did state that they had some type of adverse event, that being headache and chill, general weakness. However, they did state that this resolved in 1 to 2 days. I want to spend some time here with talking about, these trials and what did it look like for severe COVID. for the Pfizer trial, there was one person, a single person, that had a case of severe COVID. There were 10 people in the study period that had been diagnosed with severe COVID. There was one person that was diagnosed with severe COVID in that vaccination group. For the Moderna trial, I think that honestly, I want to give it a moment of silence because it's that amazing. There were 0 individuals in the Moderna trial that were diagnosed with severe COVID. How amazing is that? So that means that nobody at any point had to be ventilated. That means that nobody at any point had to be hospitalized for COVID-19. So if they developed COVID-19, it was not a serious case. So how amazing is that? So I do want to kind of harp on just the groups that they studied in here. And who were those individuals that developed COVID-19? So they did pay special attention to our elderly populations. And why is that? That's because our elderly populations are very much affected by this COVID-19 virus, right? So they had a good number of elderly individuals in that in the studies. So about 25 to 30% in each study. And then but in those cases that they saw for COVID, do you think they came from the older groups, from the older individuals? No, they did not. In those cases that they saw of the development of COVID, it came from those individuals that were younger. It was in those younger groups. For Pfizer, it was in that 16 to 55 group. For the Moderna trial, it was in that 16 to less than 65 group. Or 18, excuse me, to less than 65 group. So what does that mean? That means that if you are younger, you are not exempt. So I'm sorry, because I know that was something that's been floated, but I can attest, and even from this study trial information, that is not the case. You can in fact be infected should you be younger. So I saw the question in the chat, and I do see we have more questions in the chat. As I said, we are prepared to go a bit longer with addressing each one. But I get the question of which vaccine would you take? And do these vaccines, would you prefer the Moderna vaccine over the Pfizer vaccine? So this is my favorite question, because I prefer the Moderna vaccine, but I got Pfizer. No, I'm kidding. So with these having pretty equal numbers, so 94.1 and 95%. And when taken into consideration, what did that 94 and 95 look like? So there were 11 individuals in that Moderna study that actually developed COVID, and there was 8 in that Pfizer study. So it's a difference of 3 people. So let's put that into perspective. As far as numbers wise, they're quite similar. So I received the Pfizer vaccine, and I'm completely A-okay with that. I believe in both of these vaccines. I believe in the technology in these vaccines. They both use mRNA technology and to get the job done, and I believe in that. So what is the difference between these vaccines, if not the numbers? So the difference in these vaccines becomes the question of how they are actually made, what they're actually packaged into. So to make sure that vaccine or that mRNA reaches different areas in our bodies, or those different cells it needs to go to, it is encapsulated in what we like to call a lipid vehicle. So let's talk about lipids, right? Lipids are able to go through the cells. They're able to penetrate those hard places. So that's why they package that mRNA there, because they need to make sure that those cells get that message. So the difference between the Pfizer and Moderna vaccines are those lipid vehicles that they are made out of. That is the difference. So does that make any one better? No, it doesn't. It's just a matter of how the scientists there thought would be the best way to package that RNA to make sure that it's delivered into the cells. So what does this require? This requires different temperatures for storing. So that's when you'll hear like, oh, you have to store the Pfizer vaccine, at a colder temperature than what you would store the Moderna vaccine. The Pfizer vaccine requires X, Y, and Z. Yes, this is why. This is just what the mRNA is packed into. Is just different than the Moderna study. Does this make the Pfizer vaccine any worse than the Moderna vaccine? No, it doesn't. It's just a matter of just, how they're packaged. And that kind of is just the difference in the days in between when you can get the first dose, when you do the first dose and then the second dose. So with the Pfizer vaccine, you have 21 days that separates it. And then with the Moderna vaccine, you have 28. So that is virtually the difference. The difference is not in the numbers in terms of efficacy. If you were to receive one vaccine over the other, you would certainly be fine. So yeah, so I think that, that's my answer to that, that I don't prefer one over the other. I'm definitely fine should I receive either one.
Jacinda, can I add something?
Yes, ma'am.
You were saying? So like Jacinda was saying, it's more of a logistical concern as it relates to the fridge and how it has to be stored. You really don't have a decision as to which vaccine or choice as to which vaccine you receive. As far as healthcare workers, once the Pfizer vaccine was approved, it was approved first. They started, mass delivering the vaccine. And generally, if you see someone who received the vaccine in the last few weeks, they probably received the Pfizer vaccine because logistically that's what was sent out first. And then a week after the Pfizer vaccine was approved, well, through EUA, the Moderna vaccine was approved through EUA. And so you'll start to see Moderna vaccines being given. When I will say that essentially, if you start with Pfizer, you have to complete with Pfizer. If you get Moderna, you have to get Moderna. There's no way to swap the two because that's not how it was studied. And generally, with any drug, any vaccine, generally we use drugs and vaccines how they were studied in the trials because that's where the evidence lies. So when it becomes available to the general public, I'm not sure which one will have more in production. I think it will generally come down to what's available. And you'll, I'm sure, be told which vaccine you received so that in the 3 weeks for the Pfizer and the 4 weeks for the Moderna, you'll know which one to get as your second dose.
Great job. Thank you, Dr. Maxim. And I am going to go ahead and just elaborate more on just the pregnancy aspect, and then we are going to launch into actually answering you all's questions to make sure that we can get that stuff done for you all. So from the pregnancy standpoint, I think that it is first important, that we discuss that before different vaccines have been recommended in the pregnancy population, they had not been tested in humans. So what does that mean? That means that prior to the flu vaccine being recommended for pregnant women, that vaccine had not been tested in pregnant women. So how did they make that recommendation? They looked at risk versus benefits, right? So the risks were, if the pregnant women were to get the actual flu, that would be more detrimental than if they were to get the vaccine. So that's how they assess whether, the vaccine is something that women should get. But they also made that, but they made a caveat to that decision. So they said, yes, the risk are the benefits are definitely, greater than the risk. However, we do not think that pregnant women should get the live vaccine. And that is because, that live vaccine, that live virus may end up being transmitted across the placenta. In the event that it is transmitted across that placenta, then it could damage the fetus. So let's talk about the COVID-19 vaccine and how that's related to just kind of how we've gone about recommending vaccinations in pregnant women. So I want to blanket this with, I always, give any pregnant patients that I have in terms of the vaccine, I give them the facts of the vaccine. I make sure that I tell them that pregnancy is a restrictive airway disease. What does that mean? Pregnancy makes it hard for you to breathe. So should you be infected with COVID-19, which also makes it hard for you to breathe, that would be very detrimental to the fetus. So with that being said, the risks, the benefits far outweigh the risks, in my unpregnant opinion. However, per the and as Dr. Dio talked about, if in terms of the immunization, guidelines, per those guidelines, they also in the Association of Obstetrics and Gynecology, they recommend that pregnant women do also, and lactating women do also receive the vaccine. Why do they recommend this? Because the benefits far outweigh the risk. However, they recommend this with the caveat that the pregnant women be explained each and every risk that could each and every risk that could be associated with the vaccine, and that it be explained that pregnant women are not yet included in these studies. There is information and there are studies to be done on pregnant women as well as children. However, they have not been conducted yet. So this is also information that I provide to pregnant patients. So of course, it is your choice whether you are pregnant or lactating, whether you decide to get vaccinated, but I do want to leave just that statement. So then I also want to go into the Moderna study, which had 13 women that were pregnant. They did not know that they were pregnant at the time of vaccination, but they were pregnant during that time of vaccination. There were 7 women in that placebo group, and then 6 women that actually received the vaccine. So there was a spontaneous abortion, there was an elective abortion, and there was an actual pregnancy-related event. All of those occurred in that placebo group, so none of those women have received the vaccine. They are continuing to, follow these pregnant women, and I think that it's important that we remember with all of these vaccines, these studies are ongoing. These studies are slated to go for 2 years, meaning that we will continually see information populated in regards to these studies, and we'll continually see information, in regard to pregnant women and things of that sort. Also, Moderna also tested their vaccine in rats. And rats are often used in drug development and vaccine development, and that is because their biological systems are similar to that of humans. And when they tested that vaccine in rats, pregnant rats at that, they did not note any adverse events or any mishaps postpartum with the use of that vaccine. So as of right now, I offer that information as I have it, and I also state that there are studies and trials that are oncoming with information in regard to the vaccine and the use of that vaccine in those vulnerable populations. So with that being said, we are going to go ahead and launch into some questions. So I am going to ask the audience, my ladies, are we ready? For the onslaught. So I will make sure that we go ahead here and get these questions answered. So I guess one of the first questions that I saw was in regard to a woman asked a question in regard to her mother, who was older, and she has type 2 diabetes and hypertension, and she asked, would it be wise for her elderly mother to receive the vaccine? So somebody, go ahead and please feel free to offer a response to that question.
Brenda, I think yeah, you're on mute. There you go.
Okay, cool. If you have diabetes, and what was the other one?
Diabetes and hypertension?
Yes, because both of those are actually risk factors for having complications if you do end up getting COVID. The patients that do end up on, ventilators and they end up being on oxygen after they get COVID are those people who have diabetes, heart failure, hypertension. So I think more so important than somebody who's younger and healthier, if you do have those patients who do have those comorbidities, absolutely. Another thing that I think some of us don't think about, I may not have hypertension or diabetes, but your parents might, your grandparents might. So just kind of take those things into consideration as well when you are trying to decide whether or not you should get this.
So in terms of questions, I guess, an add-on to that question was that the mother also has periodic seizures. So would those periodic seizures kind of be a limitation for the receipt of that vaccine?
I don't see why. unless anybody else has anything else to add. Yeah, no.
I don't think it would
It
preclude.
shouldn't.
Yeah.
It shouldn't be a concern. And the reason why I would answer that way is, one, that's not, like, seizures aren't a side effect that we have seen in any trials so far. Seizures have not been reported in the upwards of 4 million people who have now received the vaccines. So again, I would consider it more in a holistic point of, if your mom, your dad, your grandmother, yourself, whoever it is, if you have comorbidities that put you at risk to have worse outcomes, meaning either severe COVID, either death, the risk of any side effects are a little more minimal than if you actually did get COVID, because again, that population of patients, or patients who do have other health conditions, are the ones who are at risk of having worse outcomes compared to patients who are not who don't have those conditions. So I would look at it more from that point of view.
Yeah, and the only group that I'm familiar with that is absolutely contraindicated to get the vaccine are going to be people who have, previous anaphylaxis to vaccines. And you guys can correct me if I'm wrong, but that's what the two of the patients in the UK who developed anaphylaxis. Those patients have had anaphylaxis to other vaccines and they carried around EpiPens. So these aren't people that, are healthy and don't have any type of hypersensitivity reactions. So if you don't fall in that category, we definitely recommend getting it.
Yeah, I will say, oh, sorry.
I'm sorry, Brenda. No, I was just going to add, also patients who, if you get the first dose and you do get a severe reaction to it, then the recommendation is to not get the second dose.
Yes. And then one last thing to add, if you actually look at the EUAs that were approved, you can see the ingredients, and it's honestly nothing that would really make you go, what is that? It's like potassium chloride, sodium chloride. So.
Yeah, and so for the other thing I will add is for the people who have had allergic reactions to the vaccines, if we look at the ingredients of the vaccine, and so I'm specifically referring to the Pfizer vaccine, but the Moderna one is pretty similar too. The ingredients of the vaccine, like Brenda had said, was potassium chloride, sodium chloride. It does have some polyethylene glycol, which is the lipid vehicle that helps it get into the cell, and the mRNA. So looking at all those ingredients, the ingredient that is likely to cause the allergic reaction is actually that polyethylene glycol. So for people who maybe have had some reaction, you might want to have that discussion with your physician, but also keep in mind that there is polyethylene glycol in a lot of stuff, including the food that we eat and the drinks that we drink, and we don't typically just have allergic reactions all day long. And so that is the ingredient that would likely cause it, but a lot of us have been exposed to it and we don't all have allergies. So I would just kind of keep that in the back of my mind as well.
So I see we have the question of, if you receive antibodies, should you still get the vaccine? If you have been infected with COVID-19 prior, is it important that you get the vaccine? And then I also saw, how long does that immunity last for the vaccine? So I think that all these questions kind of coincide. And I guess since I, began talking about that, I'll pick that question up and then I'll leave it to you all to pick up any slack. So in terms of, if you have been infected with the COVID-19, so we know that the receipt of the vaccine right now, those people that can get the vaccine is tiered, right? So you have, healthcare providers that are getting it first and the most vulnerable populations. And then we're kind of going, there. So in the event that you had COVID-19, in the event that you were infected before, it is not recommended that you go ahead and get that vaccine. not to say that you are not important, because I'm sure you were important to everyone in your life. You are not in that utmost important group to receive that vaccine. However, it is, why is that? It's because it is the idea that you should be protected for at least 90 days against being reinfected. In terms of antibodies, if you had received any type of monoclonal antibody or any type of antibodies, the plasma antibody, the convalescent plasma, if you received that, then it is recommended that you wait 90 days. 90 days until you receive the vaccine. And why is that? That's because you want to allow your body proper access to be able to mount that immune response without the influence from the antibodies, from the convalescent plasma, to go ahead and mount that immune response against that vaccine. And then I also saw if you had the flu or if you were sick, should you get the vaccine, or how long should you wait? So if you are currently not feeling well, whether it be COVID-19, whether it be the flu or something of that sort, you want to go ahead and wait 14 days. And why is that? If your body's busy fighting off the flu, then it won't be able to mount the appropriate immune response against that COVID-19 vaccine. So it is recommended that you wait 14 days before you receive the COVID-19 vaccine. I don't know if you guys have, and then we also have a question about just T-cell, about the long-term, the longevity of the resistance, and basically about maintaining immunity in T-cells and B-cells. So I will not get too much in depth about, T-cells and B-cells. What I will say is that, this is the beauty of that mRNA technology. And that is because our bodies are actually forming and forging that immune response, because our bodies are actually making that spike protein, via the message that's been delivered from that, via that mRNA. It's like our bodies making that immune response is kind of similar to if you were getting a live vaccine. So with that being said, you kind of have that same generation of antibodies that you would receive should you get a live vaccine. So kind of like briefly scraping the surface, but not getting too deep into this. With live vaccines, you won't typically need to get, what's called a booster shot or another dosing of that vaccine. And why is that? That's because that vaccine is live. Your body is able to mount an optimal immune response, meaning that both your T cells and your B cells have kicked in, and if they have made antibodies, and you have the utmost protection. When you have an inactivated vaccine, because your body's not really seeing, a live part of that, it's not really seeing the virus, in its complete viral state as it would with a live vaccine, you won't really develop those type of antibodies that you would develop should you get a live vaccine. So that's why you see, that's kind of like that T cell, B cell difference there. So the idea with the mRNA vaccine is that you will develop, that T cell and that B cell, you will develop that type of immunity. However, do we know that to be true? No, we don't. So that's why these studies are ongoing. And then they will then, check for antibody titers to see whether there is an to see the immune response that individuals will indeedly have. Go ahead, Michelle.
I just wanted to add something, not about T cell and B cell mediated. I'm not doing that, but I did want to add, as it relates to the clinical side of things, asymptomatic patients usually don't have antibodies, high antibodies when compared to somebody that has severe COVID. So kind of going back to the question of if I've had COVID before, do I actually need to get the vaccine? And 9 times out of 10, your antibodies from your previous infection have already, waxed and waned. But specifically for those asymptomatic patients, you're likely not going to have that antibody response that you would actually have in someone who was hospitalized, needed to be intubated, et cetera. So it's important to know that regardless, you should strongly consider getting the vaccine.
And I'll add that I have had patients who had COVID in March, and now they're back and they're testing positive again for COVID with symptoms and everything, going through this whole process again and having these long-term effects. So I'm those patients, I feel like my heart goes out to them because they not only experience it once, but twice. And I'm sure that those patients will be willing to get the vaccine, even though they've had COVID before. They want any protection possible. So yeah, I just wanted to add that.
Definitely.
So, Miss Jean, Kanisha Jean, I see your question here, and to reiterate, if you have received the COVID antibody transfusion, then it is recommended that you are vaccinated. However, that you wait 90 days, in between that vaccination. I think that I did see that question in regards to the mRNA and the chip. No, there is no chip with the mRNA. The mRNA technology, while advanced, does not include a chip. Messenger RNA is normally, utilized by our bodies. What it is it just delivers messages to the cells. We normally have mRNA in our body as a part of our genetic makeup. So what they did was they actually employed mRNA to just go ahead and transmit that message of making the spike protein significant for coronavirus, and then our body mounts immune response against that spike protein that's introduced via the mRNA. The mRNA is once again disposed of as soon as our cells receive that message. So that is nothing, that should be of you guys' concern in regards to the chip. So I have a question from JMM. The individual said, this is where politics and epidata clash. The epidata is negative for COVID-19. The epidata in regard to negative outcomes is relatively clear. People of color with known comorbidities. Not sure. So I guess they asked, why is there not a phase 2 candidate cluster specifically given priority to people of color with known comorbidities? The specific explicit recognition on the most affected group in the conversation of the vaccine. So I think that the gist of this question is, why are there not a significant number of Black people, included here in the actual makeup of the study, and will we be advocates of this framework? So this is a conversation that we actually had amongst ourselves yesterday. So I think that, we can go ahead and really dive into this. So whoever wants to go ahead and start, I feel like I'm taking over the convo, so go ahead.
Sorry, can I jump in? So I'm reading the question just a little different, actually. It's reading to me like he's actually talking about the prioritization for vaccinations.
Oh, my apologies then. Yeah, by all means, however you.
Yeah, like why aren't Black people at the front of the line, like Black people or minorities with comorbidities? And I can give a little bit of insight into this. So I participated in a webinar where there was a physician who was part of a group that worked on the framework for how to prioritize the vaccine groups. And for the gist of what they were trying to say is, so now in the medical world, we are starting to recognize that race in and of itself is not a reason why, like there's no biological explanation for why race makes somebody more likely to have hypertension. Racism is, right? So we need to start kind of deconstructing how we approach these things. So what is making Black people more likely to have comorbidities and more likely to be exposed to COVID? It is the types of jobs that they have. It is the fact that they tend to have jobs that don't pay as much. They don't have access to health care. And so in making the priority for who should get the vaccine, instead of placing race as a factor, which then continues to propagate this thought process of Black equals hypertension, Black equals diabetes, their goal was to focus more on the social determinants that make Black people more likely to be exposed to COVID and have bad outcomes, because then they had these comorbidities. So they focused more on congregate settings. So what types of jobs do these people have? What types of exposures do they have at this job? So are you working in a grocery store versus are you working in a hospital? Are you a nurse versus are you a police officer or an EMS? So that's what they focused on in terms of their prioritization recommendations that they made, was people that live in congregate settings. So that actually would cover nursing homes. It would cover our prison population. It would cover people who live in multi-generational families, people who are essential workers. That would cover a lot of our minoritized people as well, without having to single out and say Black people should get it first. Because again, you have to think about, we have to balance this message of, we're not trying to push Black people to the front of the line necessarily, because then that just also adds more like suspicion to the process, right? We're trying to be equitable in how we do this, while also highlighting and understanding why people are more likely to have the infection in the first place and have bad outcomes. I hope that like answered the question, but yeah.
That was excellent, Yewande.
That was great. Thanks, Yewande.
You can just, we're done.
Beautiful.
Mic drop.
I also want to discuss, and my apologies, I'm scrolling through the chat, so many questions. So we will also make sure to compile these questions so that you all will be able to, so these questions will be compiled and disseminated to us on the panel, and we will make sure that if your questions are not answered while we are on this call, that they will be answered at some point. So absolutely. And I think that, you know, we got that question of, will we have to get this vaccine every year? And I think that, you know, the best way to answer this, and you know, I hate to give just kind of this really unsure response, is we don't know that yet. So as of right now, as I said, these studies are ongoing. So they're stretching it out to 2 years. But what we want to be able to see with these studies is a development of titers. What do titers look at? How much immunity we have to these vaccines. So the way the increase in titers pretty much determines, will we have to receive another vaccination? Will we need another dose? Or will we have adequate protection, you know, against this coronavirus? So currently they do see, because there have been phase 1 and of course phase 2 trials conducted with these vaccines where they did look at this, especially with Moderna. I think Moderna did a very, very transparently amazing job with the way they laid their study out and looking at the immune response and kind of thinking about the longevity of the vaccine. And they did see a great immune response to the vaccine. They did see a high amount of titers. However, like I said, that question is just unanswered, you know, as of right now. So we will see as the data comes, and we will, you know, of course, be here to digest it and tell you guys where to go for that. We also got the question of, if you all are, I also got the question of, if you're immunocompromised, what does that look like for you getting the vaccine? So if you aren't able to mount, you know, the same type of immune response as someone that is competent, should you get the vaccine, and what does that look like in terms of vaccination?
Go ahead, Brenda.
Oh, you got it.
I was going to say, I know with the Pfizer trial, because again, that was the one that I read first and that I have burned in my brain. They did have HIV patients, and they are going to actually publish that data later. It did not come out in the original interim results. As far as transplant patients, those patients were not included in the Pfizer trial. However, this is not a live attenuated vaccine. And so ACIP, I believe, has already said that, you know, anybody, again, going back to who can't get it, is the patients who have anaphylaxis, hypersensitivity, or if you develop, you know, reactions after your first dose. So those patients should still get it, because again, they're at risk for developing you know, severe COVID. Another piece is, if you're on monoclonal antibodies, or if, you know, like if you have IBS, they're actually recommending that you take it towards the end of your course, right before your next course, which is essentially when your immune system is at its strongest, if you will, before, you know, your immune system gets knocked down again. So there is guidance out there for patients who are immunocompromised, immunosuppressed, but those patients should definitely get the vaccine as well. And we'll kind of look forward to more results as they come out for those populations.
I also see the question of Bell's palsy. So can anyone on the panel, I know that, this is our favorite question. So can someone on the panel attest and speak a little bit on the Bell's palsy question?
Yeah, I can speak a little bit about it because that was something that I was looking into specifically in the last few days. So basically, the Moderna vaccine had about 4 people of the, I believe they had the 30,000 population. So 4 people of the 30,000 that had Bell's palsy. And in looking down and like drilling down a little bit more into that data, 3 of the 4 people were in the vaccine group and 1 person was in the placebo group. So the question is, what is the likelihood of this vaccine to then cause Bell's palsy? To answer this question, I think we need to go back into like foundationally, like what is Bell's palsy and how can we make this this linkage? So Bell's palsy is basically facial paralysis. It is temporary in a large majority of people and usually goes away within 6 weeks. Usually in most cases, we call it idiopathic, meaning we do not know how it happens, when it happens, what caused it. And in the general population, typically every year, there are 13 to 34 people for every 100,000. So you're talking of about, I can't do math right now, but a large number of people that spontaneously, for no reason, we can't tie it to anything, they get Bell's palsy. So then in order to then take that information and try to figure out, did this vaccine cause the Bell's palsy in 3 people, we would have to see higher numbers than what we typically see in the general population for us to be able to make that correlation that the vaccine caused Bell's palsy. And that number is not, it is way below what is typical in the general population. So what it looks like and what the FDA concluded on based on that data is that it is unlikely that the vaccine was the cause of the Bell's palsy, and it was more likely a coincidence that those people got the vaccine and then had Bell's palsy, because we typically have cases that spontaneously happen for no reason, and we cannot tie it to anything in a normal world. And I hope that like kind of helps answer that question.
Thanks, Yewande. And as we are winding down on that 2-hour mark, I think that we can spare an additional 5 minutes after. But I see the question, and I think this is important, to answer. Black people tend to have lower white blood cell counts in the normal range. Does this in any way impact the efficacy of the vaccine? So I think that, once again, because I ended up, I guess I was one of the people that went over just like the vaccine and what we saw in the trials. I think it's important that when we think about this question, we think about what did the actual study show? So while yes, only it was 9% and 10% in the Pfizer and Moderna trials respectively. However, I would like to note that those individuals that actually developed COVID, they were not Black. It was not Black people that developed it. So what does that mean? That means that vaccine was indeed efficacious. That meant that Black people were indeed able to develop those white blood cells necessary for them to mitigate that virus and for them to make sure that they were, for them to be protected, they were able to develop the adequate amount of antibodies. I am also not absolutely scientifically sure that is correct. So what I could do is get back to you once I have data to support that answer. However, and I think that it's important that we also consider, what does normal range actually mean? So normal ranges vary between hospitals. Normal ranges vary between actual documentation. Normal values vary between institutions. So I think that it's important to really distinguish what it is that you mean and what it is that we mean by normal range, because by all means, Black people should be able to mount an appropriate response to any disease and any type of vaccine in terms of white blood cells. But in terms of the numbers, which is where I think, this is the best way to focus this, is that Black people were not those that were actually infected with COVID-19 in these clinical studies. It was not Black people that were. So with that being said, and what does that tell us? That tells us that the vaccine indeedingly did its job, and that Black people were indeedingly able to form the appropriate response necessary. So I see additional questions. There are a lot inside of the chat.
There's one question that I think is very important.
Yes, ma'am.
No one has died from getting the vaccine, not a single person. 2 people died, I believe, in the, I'm mixing them up now. I can't remember if it was in the Pfizer or the Moderna one. One of them was from a heart attack that was unrelated to the vaccine, and the other one was from an arrhythmia.
It was from the Pfizer trial.
Yeah, it was the Pfizer, yeah.
Yeah.
No one has died because of the vaccine. But many people, over 300,000 people have died now because of COVID.
Just in the US.
Yeah. And sorry, there was one question where someone was asking about why 9% of the population in the study was Black and how that is okay. And I don't know, Jacinda, do you want to talk about that?
Yes, I can talk about it, or someone else can. I don't want to go ahead. I don't want to
I think you did touch upon
monopolize the conversation. But yeah, I think that Yewande actually did a beautiful job at describing this, essentially describing this point. And so I'll read the question and just try to dig into what I feel the question is asking. So with us being the minority, so I think that it's important that we actually look at, the percentages and the groups that were actually tested in the study. So while African Americans were, and these are studies that people have the opportunity to enroll in, right? So they ask them to enroll. So it's not, you're going to be in this study. No, they are given the opportunity to enroll. So once again, we kind of have that factor of, who are the individuals that are enrolling in the study. But I think that there's a different way that we can kind of look at this is, yes, we were afforded, this vaccine prior to elderly people or the vulnerable population. But let's not forget that we are also a vulnerable population. As Dr. Dial stated, socioeconomic factors and systemic racism and social constructs will have kept us a minoritized and vulnerable population. So we are a vulnerable population, so we should have been represented there. And we are represented there in an amount that is close to what we represent the entire United States population. So I don't think it's, a matter of it being afforded to us before the most vulnerable. I think it's being afforded to us as well as elderly individuals, because we all fall underneath that vulnerable category. But I think it's also important to note that elderly individuals also comprised about 25 to 30% of those individuals that were included in the study. So what does that mean? That means that they specifically stratified, meaning separated these groups so that they can look at older individuals and that they can see how the vaccine actually performs in those older individuals. So I think that is important. So the same way that they went through the processes, and Moderna stopped, their clinical study to make sure that Black people who is a vulnerable group is accounted for in their study is the same way that they made sure elderly individuals were accounted for. And then those individuals that had, other comorbidities were accounted for. And some of those individuals being overlapping, right? Those that are Black, those that are elderly, and those that have comorbidities. So some of them kind of filling both of those roles. And that is because, they wanted to make sure that this vaccine was indeed effective in these vulnerable populations.
I think if you also look at some of the other trials, so I wanted to see, well, what did other flu vaccine trials include in terms of the Black population? What about HIV studies? So although there were about 5 vaccines that got approved for the H1N1 influenza pandemic, some of the studies don't even publish the information, right? But one study said that there were actually 11% Black patients in that study compared to 85% white patients. In HIV studies, the very first idovudine antiretroviral for HIV, there were 7% Black patients in that study compared to 49% white patients. And then in a more recent HIV study, there was about 12% Black patients. So it's pretty comparable to, again, what we've been saying all night, that this kind of reflects the current population. And so it's not, yes, it is a small percentage, but when you look at other studies, it's about the same. So I think both studies did a good job of recruiting the amount of Black people that were in the studies.
All righty. So with that last comment, we are going to end on a note of, we hope that this program was informative for you all. It is our hope that we have given you a bit of solace and a bit of education as you all go forth and make that decision to be vaccinated. I would like to ask that you all follow us at Pills Connect on Instagram, Facebook, Twitter, LinkedIn, and on YouTube. This conversation will be recorded, and it will also be placed on YouTube. We will also be sure to address any questions that were missed throughout this informational session. We bid you all farewell. Please stay safe, and Happy New Year.